Okay, but it's now, but don't I have to do it with this?
Yeah. So then what you can do, you probably just go into full screen.
Okay, so these are your targets for today. We're going to be ultimately using models and amino acids, but before we do that, we have to do yesterday's targets. We're going to relate the structure of function to phospholipids and other lipids. And we're going to be using models to identify polar and nonpolar parts of phospholipids. And then after that, we're going to go to today's targets, which are to diving into amino acids, which are protein derivatives. Or amino acids are monomers of proteins. And we're going to use models to help look at hydrolysis and dehydration synthesis. Okay. So we talked about single bonds and double bonds in the fatty acid chains. And single bonds are unbent, and they're solid at room temperature, and they're tightly packed. And we have double bonds that are bent because they have less hydrogens, and they're liquid at room temperature. And we looked at fats. Those are types of fats. Now we're going to look at steroids. And the molecule on your left is a cholesterol. And that's a type of fat. And the molecule on your right is a derivative of cholesterol. It's found in high amounts in males, especially adolescent males. That's testosterone. And if you could make one little change to that testosterone molecule by adding a CH3, and that would make it estrogen. So right there, if you just put a CH3 on there, it would change the whole property of that hormone. Cholesterol makes our sex hormone, so without it, we can't do that. It's one of the reasons why people who have eating disorders stop menstruating or having hormone cycles because they don't have enough fat. But all these steroids are fused rings of carbon, so they look different. But they are just like any other lipid. They are nonpolar. And if you notice, they really have very little oxygen, and that's what makes something super polar. Yes, there's a little bit of oxygen there, but it's not enough to make it polar. So these are nonpolar molecules. One of the most, we're going to take a look at another class of lipids. Sorry. I'm recording this after the fact because I forgot my headset, so sorry. We're doing double work. And my poor boys are sitting here waiting. What happened to Von Vic? Did he? Okay, so we're going to look at, these were the fatty acids. We looked at cholesterols, those rings, and now we're going to look at phospholipids. And phospholipids are exactly what they say they are. They have a phosphate group. So highlight the phosphate group in pink if you can. Take a second and highlight in a separate color. And now highlight your fatty acid tails in yellow. Sorry, are you guys bored? Class, these four young men are being wonderful putting up with me recording this again. The long chains, let's do the fatty acid chains in yellow, highlight in yellow. And this is your fatty acid, that's your lipid part, and the phosphate is your phospho part. What's interesting about this is it has two properties. It has a polar region and a non-polar region. So take a second and circle the polar region and put a box around the tail region. Okay, here's your polar region. See, it's got a positive there. Hopefully you can see that. And it's got some oxygens, which are negative. That makes it really polar. And here's your non-polar part. There's only carbon and hydrogen. And that makes it super interesting because phospholipids make up every single membrane of every cell in your body. And even non-humans, all animals, all plants, they all have cells. You know, cells all over your whole body is made of billions of cells. Every single cell has a membrane around it, and it's a double, what we call lipid bilayer. And there's a double layer of phospholipids. So what I'd like for you to do on that picture there in the circle, which shows the cell, inside the cell. So there, if you see, there's polar heads and non-polar tails there. Inside that cell is water, and outside that cell is water. I'm a little ahead of myself, sorry. Inside and out of the cell is water. So there's the inside. And there's the outside. And you will find water in the cell and out of the cell. So what I'd like for you to do is in pink, label the heads, the polar heads in pink on this, and label in yellow the tails on this picture. So get out your pink and yellow and relabel it. Take a second to do that. Remember, the heads like water and the tails don't like water. So polar parts, polar dissolves polar, right? Non-polar likes non-polar. So if you're going to highlight the heads, which are polar, the pink, let's do it. Give you a second to do that before I show you. Boy, I give you a lot of time, don't I? You better be working on this. So the pink are the little circles. That's the polar part. You'll see those heads on the inside and outside of the double membrane. Notice how there's two layers, right? And then the non-polar part is that whole, all those tails facing each other. So the tails flip in to face one another because they don't like water. They're going to get away from the water. And the heads like the water, so they're staying on the outside. So the cool thing about this is that these tails are probably saturated and they're pretty straight. But depending on the environment, you can add double bonds to those tails and make them a little more fluid, like in cold regions. You know how butter gets really hard if it's cold? Maybe you want to add a couple double bonds here so that it'll stay a little more fluid in a colder region. It may be an adaptation to help it survive longer or better in a colder environment. So that's our work with lipids. So please take a chance to answer questions 1 through 5 in the next couple pages, and then we'll resume with amino acids. How do I stop this? Do you know? Right here?
Over here.
How do I stop it?
Uh,
This one?
Yeah.